Good morning students. So today we have to start the one more concept in the genetics, that is the gene mutation. Okay, already we completed the chromosomal mutation, or we called as the chromosomal aberration. So mutation is nothing but the sudden change occur in the hereditary material, means it may be DNA, RNA, chromosomes, okay, or it may be gene. If the sudden changes occurs, that we called as the mutation. So chromosomal mutation is there and the gene mutation is there. Chromosomal mutation means changes occurs in the structure of the chromosome or the number of the chromosome. Okay, so one more is the gene mutation, means changes occurs in the structure of the gene or composition of the genes here. So mutation is nothing but the sudden change occurring in the hereditary material is called as mutation. It may be harmful, it may be beneficial. Mutation may be harmful or may be beneficial. So DNA is the highly stable molecule that replicates with amazing accuracy. Some errors of replication do occur, means DNA, which is the highly stable molecule, so due to sudden replication occurs, that's why if the error occurs, then also replication is going on in the cells. Okay, so how it occurs and what are the changes occurs in the genes, chromosomes, that we have to study. So it includes the change in the gene structure or composition, that we called as the gene mutation or point mutation, and the changes in the structure, changes in the chromosome, either in the structure or in the number, that is the chromosome mutation. So the term mutation, who coined it first time? So you have to see the history of the gene mutation. Okay, so you know the scientists Hugo D. Rice, T.H. Morgan, H.J. Muller, all these are worked in the field genetics, and even Mendel. Okay, so the scientist known as Hugo D. Rice first used the term mutation to describe the sudden heritable phenotypic changes in evening primrose, means that is the flower plant, where the changes occurs in that flower. He did the study in that only. Okay, and white-eyed Drosophila in the population of red-eyed. Here also mutation occurs. You studied chromosomal mutation in that also. White-eyed means occurred in the wild red-eyed flies. Okay, so you studied that in sex-linked inheritance. Again, Muller first used X-rays to induce mutation in fruit flies. Like these, different scientists worked in different plant species and different animal species. Okay, so next, which are the types of the mutation? So here mutations, or we call as the change in genes or chromosomes. First I told gene mutation, second I told chromosome mutation. Okay, and here again replacement of one nucleotide by another, addition or deletion of a nucleotide. So these will be comes under the gene mutation. And the second that we called as the chromosome mutation. So here what will be occurs? Which types are there in the gene mutation? So change in the number of chromosomes, that is the polyploidy, aneuploidy. So 43 number chromosome, 42, 45. So like these, those we called as the polyploidy, aneuploidy, and change in the structure of chromosome. Okay, these two types will be comes under the chromosome mutation. So next is the first, that is the gene mutation. So a gene mutation is defined as an alteration in the sequence of nucleotide in DNA, means changes occurs in the nucleotide sequence. Nucleotide means it contains the amino acid like AUG, methionine, alanine, lysine, different amino acids, those we called as the nucleotide sequence which is present in the DNA. So changes occurs in the sequence of nucleotide. So this change can affect a single nucleotide pair or larger gene segment of a chromosome. Means it may be sequence of nucleotide, it may be in a pair or single or larger gene segment in the chromosome. Okay. So in case of the gene mutation, again different types are there, subtypes. So first one is the point mutation, frame shift mutation, base substitution mutation. Under these subclass, again different types will becomes. Those are silent mutation, missense mutation, nonsense mutation. In case of the frame shift, insertion and deletion. Okay. So last one, that is the transition mutation and the transversion mutation. So first we shall see the point mutation. Point mutation means what? So point mutations are the most common type of the gene mutation. Means you can able to see the much common, that is the point mutation, also known as the base pair substitution. Okay. So AUG, A means adenine, guanine, cytosine, thymine. Like these nucleotide sequence will be there. These will be code for the particular amino acid. Okay. Then here again three main types they are given. Silent mutation. Okay. The change in one codon for an amino acid into another codon for that same amino acid. So you just see this GAA, here again GAG. So here also glutamine, here also glutamine, means same amino acid is there, but nucleotide sequence, that is the base pair, here it is A, here it is G, that we called as the silent mutation. Okay. So silently it means changes occurs in that base pair or in the nucleotide sequence. Missense mutation, the codon means these CA, okay, those we called as the codon. One codon means three CAT codes for one amino acid, like these will be comes here. The codon for one amino acid is changed into codon for another amino acids, means completely it will be changed. If upper side one amino acid that was replaced by the one more amino acid will be comes and sit there. Okay. So that is the missense mutation. If upper side glutamine is there, that will be removed by the another amino acid, lysine. Like this we called as the missense mutations. Okay. So last is the nonsense mutation. The codon for one amino acid is changed into a translation, termination or stop codon. Okay. So one amino acid that will be changed for the means different amino acids will be comes here. Like you know the methionine. Okay. One codon is there that we call as a nonsense codon because it don't have any work. Then next type in case, second subtype in case of the gene mutation, that is the frame shift mutation. So this type of mutation occurs when the addition or loss of DNA base changes in a gene's reading frame. Okay. So it may be addition or it may be deletion of the DNA base. Okay. Base pair. A reading frame consists three bases, each code for one amino acid. I told AUG. These are the three spellings, means three base pair codes for only one amino acid. Again three codes for one amino acid. Like these codons are there. A frame that may be, means in case of the frame shift, the codon may be changed. It may be deleted or it may be addition will be takes place. A frame shift mutation shifts the grouping of these bases and changes the code for amino acid. Okay. The resulting protein is usually non-functional. So amino acid sequence means again proteins only. So if the changes occurs, those proteins or the amino acid don't have work here. Okay. Those we called as non-functional here. Insertion and deletion can all be shift mutations only. Okay. So here the frame shift mutation occurs. This is the original DNA code for an amino acid sequence. Here frame shift of one DNA base results in abnormal amino acid sequence. Here one is removed. Okay. So insertion or deletion will be takes place in the frame shift mutation. So first one is the original, second one is the abnormal. So insertion means what? An insertion changes the number of DNA bases in a gene by adding a piece of DNA. Okay. A small piece of DNA will be added, that we call as the insertion. As a result, the protein coded by the gene may not function properly. If the insertion takes place, the added amino acids that will be not work properly. Okay. So we called as it's a abnormal. Then deletion. Deletion means one amino acid will be removed or gene segment will be removed here. A deletion changes the number of DNA bases by removing a piece of DNA. Piece of DNA removed means of course codons will be removed or the amino acids will be removed here. Small deletion may remove one or few base pairs within a gene. Okay, within a gene means a small segment of DNA, that is the gene, which it may be consist of one or few base pairs. Okay, base pairs means AUGT like this. Larger deletions can remove one entire gene or several neighboring genes. Okay, small deletion is there, large deletion is there. Small deletion removes the one or few base pairs, large deletion removes the several neighboring genes. The deleted DNA may alter the functions of the resulting protein, means finally protein or amino acid will not form properly. So base substitution mutation. So that may be transition mutation or transversion mutation. Base substitution are mutations in which one base pair is replaced by another. Okay, so transition here, purine first pyrimidine. Okay, so a transition is the replacement of a base by the other base of the same chemical. A GAAG like this will be takes place. Transversion mutation means is the opposite, the replacement of a base, one chemical category by a base of the other. Purine is there, pyrimidine. Pyrimidine is there, purine. Means in case of the transition, same chemical composition will be comes there, purine, purine, pyrimidine, pyrimidine. Here purine, pyrimidine, pyrimidine, purine like this. Molecular mechanism of mutation. How it will be takes place? Spontaneous, induced. Spontaneous means that we call as sudden it will be occurs. Tautomerism, depurination and the deamination. Induced means physical mutations, that is the radiation, ionizing, non-ionizing, heat, chemical mutations. Due to all these agents cause the molecular mechanism of the mutation. Up to the molecular level mutation occurs. Deamination means removal of the amino acids, like these will be takes place up to the molecular level. Spontaneous, that we have to study first. So spontaneous mutation are those which arise naturally. Okay, mutations arise naturally, not through the action of a mutagenic agent. They may arise through errors in DNA replication. If the normal DNA replication is going, anything happens, if anything error is there, then what happens? Spontaneous mutation occurs. Okay, so it is not the induced. Spontaneous mutation, again depurination, deamination, tautomerism, like these different types comes under the spontaneous, or we call as the natural type of the mutation. Induced, ionizing, radiation, non-ionizing, heat, chemical mutations, all these we called as the induced mutation. During its lifetime, an organism may be exposed to variety of physical, chemical and biological agents, okay, which cause the genetic material mutation. So physical mutagens. Mutagens means which cause the mutation. Those agents we called as mutagens. So radiation is the mutagen, in that ionizing radiation, non-ionizing radiation. Heat means break the N-glycosidic bond in DNA. Glycosidic bond is present in DNA, that will be break due to the heat. Then result form a purinic site or baseless site. Base pair will be altered there. Chemical mutagens means alkylating agent, intercalating agents, like these will be base analogs. So different chemicals cause the mutation. Effects of gene mutation. What will be happens if the gene mutation will be takes place? What are the different effects? Some of the beneficial is there, some will be harmful, and some will be neutral there. So which are the beneficial effect of the gene mutation? First, produce new version of protein. Means due to the mutation, different proteins will be produced, that we call as new version of protein. Essential for evolution. If the changes mutation occurs, then the new organism, new species will be formed. Enhance the survival and reproductive success. Okay, this is the third one. Then last one, turn off harmful genes. If the harmful genes are there, if the mutation is occur, that will be removed. Harmful gene will be removed and the new one will be formed. That is the beneficial. Harmful means protein malfunction. The proper protein will be removed and the another protein will be come and sit. That will be called as the abnormal because it will not work properly. So sometimes we called as it is the harmful. Protein malfunction will be occurs. Protein will not work properly. Lethal to life. Sometimes the means gene mutation if occurs, that will be lethal to the organism. Means organism can't able to survive. Sometimes stops the development of fetus. If the gene mutation occurs in the fetus, that can't able to develop properly. Cause genetic disorder. Neutral means not significantly harm or benefit body. So genetic disorders occur due to the mutation. These we called as the genetic disorders due to the gene mutation. So see the diagrams. So carrot means how it is developed, brain. So babies how it will develop, leg, eyes and the face. Okay. So these will be occurs due to the gene mutation. Okay. So if you have any doubt, you can ask in the class or you can ask in the means you can message. Okay. Thank you.